This one seems like I don't know. This did not fully come from ChatGTP this time, so
This is a good one.
it came from my head this time.
This is great.
Welcome to Absolute Genius, a podcast series from Thermo Fisher Scientific. I'm Jordan Ruggieri, and alongside Christina Bowens, I'm thrilled to welcome Dr. Jesús Mingorance to the show today. Jesús is a scientist in the microbiology department at Hospital Universitario La Paz in Madrid, Spain. In addition to researching genomics and antibiotic-resistant bacteria, he also does a lot of work with evaluating tools, including the ones we love to talk about on our show. It was amazing to hear about his career journey and his current research, and we hope you enjoy. We began by asking Jesús to explain why tools such as ultra-sensitive PCR are so helpful for his work.
Well, there are several fields where the ultrasensitive PCRs might be useful. In the field of HIV, it's mostly when you are doing some research and you want to measure the virus or the proviral load, no, not only the load of virus, but the provirus. You look for DNA, viral DNA integrated in cells. And another one is in some group that are suppressed with undetectable levels or very low levels of virus. But for some reason, you want to monitor how are these levels going on. And these loads are usually below the quantification level of the standard techniques. qPCR has a quantification level of 20 copies per ml. Or in some cases, you want to monitor how is it going around this level, between 20 and 10, 5, and so on.
Are there, looking at the, let's say, provirus, or looking at those levels of DNA and really maybe smaller amounts of DNA, what are some of the implications of looking at that? Are there maybe presentations of those amounts that you're looking for? Are you looking just to see the presence of that virus and detect that?
Well, that depends on the project. We did, for example, one project was not only to detect, but to sequence that clones, because we had some group of subjects that were supposed to have resistance mutations, but were not treated, and but they were suppressed. So we were curious about what's happening, why the virus is not increasing that one. So we have to isolate single clones, identify those clones, and sequence them to detect whether they had mutations or not. Maybe you are measuring uh, 20 copies per 10 to 6 ppm cells, but are these 20 copies functional? So in some cases, we found that most of these copies were hypermutated, so probably non-functional.
Interesting. I'm curious to dive into maybe your recent publication with your collaborators and hone in a little bit on, looking at ultra sensitive PCR and digital PCR for that publication in particular, right? For HIV-2. Can you talk a little bit about the aims of that publication and what you were trying to accomplish?
In this particular case, this was the subject was already diagnosed, so we knew that he had HIV-2, and I think he was not responding correctly to the drugs. But when my colleagues at the microbiology department did the NASVA assay, the biomeria assay, they couldn't find any signal. They couldn't detect the virus. So they send some samples to a laboratory in France, and in France they could detect it, and even they could sequence it. So they were curious what was happening. They thought that maybe this virus was highly mutated and probably was not being detected by the NASBA assay because of this. So they asked us to test something different. At that time, we had the machine, the PCR, absolute Q machine. We were testing for other reasons. So we contact Thermo and ask them for a master mix for RT, reverse transcription PCR, and they send us a beta. So we tried with a Tachman assay, HIV-2 Tachman assay, to detect it, and it was great because we could detect and quantify the virus in this patient. So our colleagues say, okay, so we have some samples in the freezer. We could try if you can detect also with this system, because there was no other system at that time to quantify HIV-2. While we were doing this work, Altona commercialized PCR, qPCR assay. So at that time, we could test both in parallel, the Altona qPCR, the Thermo dPCR, uh, and in parallel with the classical NASBA assay. And well, those were the results. We were initially trying to identify the viral load in a single subject, but then it became an evaluation.
I was looking through your publication and I just wanted to get so this topic of, looking through cell-free nucleic acids to identify viral load. When you're using something like dPCR or even qPCR to quantify the viral load, is it very important to know the exact viral load, or is it more making sure that you have good limit of detection and confidence around that?
Yeah, usually in microbiology, precision is not important. I mean, you want to know the order of magnitude. You want to know if you have 100, 1,000. But generally speaking, it's not precision is not important.
Yeah, it does kind of remind me a little bit about, to some degree, in oncology, you're looking for circulating tumor DNA commonly, and that's always a question I love asking is, does that quantity actually matter, or is it the change or variance? And I think your response orders of magnitude makes a lot of sense there. So in oncology, which is where I'm familiar with working a lot, we know like SNPs and rearrangements are like those critically important like biomarkers that we're looking for in cell-free DNA. When you're looking, at some of the work you're doing, especially for, disease research, what types of targets or markers are most common that you're like actually designing the assays for? Is it for like transcript? Is it for specific bacterial sequences? What's the most common?
We basically look for genomes. We look
Okay.
for the presence or absence of a pathogen.
Okay.
If it is a RNA virus, we look for RNA. We'll do RT-PCR. Or if it is a bacteria or fungi or DNA virus, we look for DNA.
So do you find yourself doing a lot of custom design of primers for qPCR and dPCR? Or, if something exists in a big menu, is that, perfectly reasonable to go ahead and test? I just wonder about, the availability or complexity of designing some of the assays that you're looking at.
Most of the assays that we are doing are for qPCR. We had this opportunity of having the machine for, I think, almost 1 year. And so we did several tests on cytomegalovirus, on hepatitis B virus, and this HIV business. And we did something on bacteria too. And in any case, it's much more easier to design qPCR assays.
I'm even curious, even going back, even one more step for our listeners, why would you even need to monitor viral load?
Well, viral load is a marker of disease. All the work that we do is for clinicians. Of course, we have some research projects, and in this case, maybe there is a big role for the PCR and for other more sophisticated techniques. But most of the work of the laboratory is for clinicians. If they find it useful, then we do it. And viral load in HIV is a marker of disease progression and response to antiviral drugs.
It makes sense. Do you see some resistance even with viral strains, maybe mutations that stop certain drugs from working as effectively or evade some of the drugs? Is that something that you often see?
Yeah, this has been a constant in HIV during at least 10 or 20 years. Uh, Companies used to commercialize new drugs, to test new drugs, to put new drugs into the market. Subjects take these new drugs and then it work for a while and the virus develop resistance. So this was the origin of the triple therapy, try to avoid the virus escaping from the drugs. And with the years and with the new drugs, I think now that the level of control is very good. There is almost no resistance.
You develop methods for clinical research. So, what types of tools and technologies do you have on the bench at your disposal when you're working through those, researching those new tests that could potentially go to clinic?
Well, the main tools are PCR, qPCR, and sequencing, which is a massive sequencing, Sanger sequencing some years ago. Now, genomic sequencing, in particular, nanopore sequencing, which is very friendly and quick. And easy. But other technologies too. We are now trying to introduce proteomics, but that's difficult because we have the MALDI-TOF for identification of bacteria. This is very easy. This is not research. This is already established. And we are trying to go further. But usually, there's one thing that you have to keep in mind when you work for clinicians is that they like works to be do in a simple way, quickly, and cleanly. It's difficult to be better than qPCR, or qPCR is very well developed now. We have a lot of knowledge, tools. It's a robust technique. So it's better. It's difficult to go further.
As you were, looking at digital PCR, we love digital PCR here on Absolute Genius. So we love everything, but I love to talk about digital PCR. Did you find any specific advantages of using that technique, say, over some of the other tools that you have and techniques that you have?
Well, I have to say that I love digital PCR. Okay, I'm a biologist, so when you explain the technique, when you see the principles, so you say, "Wow, that's beautiful." So the PCR is very interesting when you don't have a standard technique for a reference or a standard for calibration. Then you can do a quick quantification even if you don't have a calibrator. Or you can use it to create your calibrator. And then you can translate this to the PCR, to qPCR. So it's I mean, maybe this is the more clear advantage.
I could imagine some advantages of either not having to use some type of calibration like a standard curve, right? Or to be able to use it to make sure your standard curve is accurate before you go
Mhm.
into maybe more well understood qPCR quantification techniques.
Yeah, not only you don't depend on the efficiency of the PCR.
If you could pick one thing that you think DPCR would be really great at, hands down, it would be the best option. What application would you say fits that bill?
Well, in our particular case, we are now part of the European network of reference laboratories, which is a new network which has been created for testing commercial assays, and so. And we are going to need standards. We are going to need to calibrate them. And the PCR would be an incredible uh, technique for this particular application.
Yeah, for that like base standards calibration. What do you think could help? Because we've talked a little bit about how, qPCR is the gold standard for many, many labs. It's really well accepted, which has been a challenge in, moving dPCR to that next level. What do you think could help propel dPCR to that next level, being like I don't know, the next gold standard of testing?
But for microbiology, we will need to explore the DNA extraction protocols because this is a strong limit. If you start with a half milliliter sample and you want to compare this with a blood culture, for example, that you are studying with 10 milliliters, you are in disadvantage. So we need some DNA extraction systems that are able to manage larger volumes of samples
Mm-hmm.
so that you can put a largest amount of DNA. Because in the case of microbiology, it's different from genetics. Maybe in genetics you think, okay, I'm going to put 10 nanograms of DNA, and this is 10 nanograms of human DNA, which is your target. But in microbiology, if you put 10 nanograms of DNA, you don't know how much pathogen DNA is there. And if you are looking for in the lower limit, so maybe you need to increase the volume without increasing the inhibitions. Development of better systems for extraction, DNA extraction, would be important.
Yeah, I could see that being super impactful. And then if you have those higher volumes, since dPCR is relatively more tolerant to inhibitors, it could be a really good platform to make sure that you get the sensitivity without losing a ton of the assay efficiency problems that you could run into.
And then if you could increase a little bit the dynamic range, would be great. In many cases, for example, when you do the standard HIV viral load detection, quantification, okay, you have subjects that are on treatment, they are suppressed, they have less than 20 copies per milliliter, so no problem. But when you have a sample from a subject that has been just diagnosed, you may have 1,000,000 copies per milliliter, 500,000 copies per milliliter, and you are over the limit for dPCR. So some increase might be interesting.
Okay, yeah. So you're saying increasing the concentration detection limit actually on the upper side rather than the lower
Yes,
side.
yes, because it saturates quickly.
Makes sense when you are working with an unknown, right? Like you had mentioned,
Yeah,
right? If
sure,
you're
sure.
on the microbiology side, you get a sample, you have no idea how much virus might be in it. So it makes sense why.
Then when you get a saturated sample, then you have to do dilutions to guess which amount is. And in particular, when you are working with bacteria, the amounts used to be larger, so it saturates very easily.
Excellent. I have another question that takes us back many steps, but I think would be important in the storytelling for the podcast episode. We talk a lot about HIV-2. Can you talk a little bit about the differences of HIV-1 versus HIV-2 and any types of implications or considerations when examining these types of viruses?
Well, as I said before, HIV-2 is mostly limited to West Africa. So for some reason, it's not epidemiologically, it's not global. Clinically, it's somewhat slower, but the end is the same, it's AIDS. And the treatments, I think, are the same, must be the same. The response to the treatments is the same. So that the main difference is epidemiological, it's geographical.
In the paper, I know that there was a line that said there's traditionally a lack of, readily available commercial assays for HIV-2. Is there a specific reason for that? Or
Well, because we don't have much HIV-2 in Europe or in USA, so companies have not there, and probably there is not an international standard.
You also mentioned towards the beginning that you work a lot on antibiotic resistance bacteria. Is that anything that relates to any of the techniques we talked about here, or anything that might be similar?
We have been doing some research. We have a problem in our hospital, as in many other hospitals around the world now, with carbapenemases. So we have bacteria that are resistant to carbapenems, which are antibiotics that are used usually in hospitals as a last line, and have most of the cases are Klebsiella pneumonia, which is for some reason has adapted very well to the hospitals and to the carbapenemases. And when you have Klebsiella carrying a carbapenemase gene and enters into the hospital, it explodes and creates an outbreak. So, and there has been a lot of work in the hospital, a lot of effort in trying to contain or to eradicate these carbapenemases, and we couldn't. Has not been possible. We have, indeed, in our hospital, I would say it's endemic for the Klebsiella pneumonia carrying carbapenemases. So we wonder why is this so, and we started to measure the bacterial load, the Klebsiella, carbapenemase producing Klebsiella load in intestinal samples from colonized subjects. Is not sick, but is sick because it's in the hospital, but it's not, the Klebsiella is silent, it's part of your gut microbiome. Is silent, does not create any problem, but sometimes it may go out from the gut and create an infection or transmit and create a second, which is colonized. No, uh, we try to understand these points and start measuring by qPCR the amounts of Klebsiella, carbapenemase producing Klebsiella in the guts, in the microbiome of the subject. So we develop some protocols. We wanted to compare the Klebsiella population with the total microbiome population. So we did qPCR directed to carbapenemase genes and qPCR direct to 16S RNA genes, which is a proxy for the bacterial population. We found that some of these patients have only Klebsiella or mostly Klebsiella in their guts. So probably they have been receiving some antibiotics that swept away all the microbiome, and only the resistant strain could survive, and most of their microbiome is Klebsiella, and that's the reason which is so difficult to stop this from transmitting to other subjects, because any small contamination, any small fomite can transmit loads of Klebsiella. So that's and we did by qPCR. It may be done, could be done by dPCR, maybe, but in this case, the dynamic range is important because we are working with samples which have high loads of microorganisms.
Are you looking to precisely quantify targets with digital PCR without a standard curve, measure viral titer, or quantitate residual DNA? Are you looking to scale your dPCR workflow while maintaining a simple and efficient process? If so, Applied Biosystems QuantStudio Absolute Q Auto-Run dPCR Suite is for you.
The QuantStudio Absolute Q Auto Run dPCR Suite is an automated, easy-to-use dPCR solution that helps improve throughput and utilization for absolute quantification of your samples. The system enables efficient utilization with multiple users able to load and schedule runs, along with greater productivity with automated plate loading and run scheduling with a single click.
It also enables consistency and multi-plate analysis so you can analyze batch sizes that match your experimental needs together for improved reproducibility.
You can learn more at www.thermofisher.com/autorun. That's www.thermofisher.com/autorun. The Applied Biosystems Quant Studio Absolute Q dPCR system is for research use only, not for use in diagnostic procedures.
And now back to our conversation.
Awesome, Jesus. We are at the career corner portion of our Absolute Genius podcast episode, where we talk a little bit about your career, how you got to where you are. Can you give us a little background on your career and maybe some of the educational and kind of key milestones for you as you went on your career journey?
Yeah, well, I would say that this has not been a typical career. I studied biology in Barcelona, and then I moved for a while to Japan, to Okayama University, and did a master degree in genetic engineering and bacterial genetics there. Then, well, I happened to knew a lady there from Madrid, Spain, not from Japan. And so I came back to Madrid, not to Barcelona. And then I did my PhD here. And I entered into in a biochemistry laboratory. I had a background in bacterial genetics and genetic engineering, and they wanted to do site-directed mutagenesis, some liver enzymes. So we cloned and we did mutagenesis. And that was great because I learned a lot about proteins and enzymes, but I miss bacteria. So I wanted to go back to microbiology. So after PhD, I went in the laboratory of Professor Miguel Vicente at CSIC here in Madrid too. He had a strong background in bacterial cell division. So I carried my knowledge in bacterial genetics and enzymes into bacterial cell division. For a while, it was fun. I learned a lot of things, and we had some projects on antibiotic resistance at that time. No? It was a kind of paradigm. You have to study antibiotic resistance. But I was not very happy because I couldn't really, I did not know the extent of the problem. So how important is really antibiotic resistance? I can read some papers, but I wanted to know how important is. So I moved to Hospital La Paz. I thought I will move for a while, just for 3-4 years. And it's already 18 years. But I'm still learning from clinicians, and I think I know something now about antibiotic resistance. And in this time, I tried to bring my knowledge on molecular biology and genetic engineering to the diagnostic field.
What a journey.
Yeah, that has been funny.
I want to study in Japan. That sounds amazing. You mentioned that your career pathway has not been traditional in normal sense here. Are there any key milestones or things that have happened that maybe have changed the trajectory of your career? And what have you learned from some of those?
When try to remember, sometimes I am surprised that I remember when I decided that I wanted to do biology. And it's strange because I had at high school, when you have to decide between science and humanities, I decided to go to humanities. So I studied humanities, and then I stopped there. I didn't enter the university. I went to work around, and one day I was reading a book, a science communication book. I read something about DNA, and I was impressed. I said, okay, I cannot believe that I have studied this at high school. It didn't impress me. I couldn't believe this. I want to study this. I want to go further on this. And then this is when I decided to study biology. And this has marked all my career. And mm, one of the tools in my career, the most important tools, has been sequencing.
That's so funny. That's almost exactly what happened to me as well. I was in high school and I was like, definitely want to be a graphics designer.
Mhm.
And then I took an advanced molecular biology class just for credits, and I was like, wow, I
Yeah,
cannot believe
yeah.
that I didn't like this. And I have loved it ever since.
I think too, it shows that you can always change your mind, right? And that's okay,
Mhm.
to enjoy things that you maybe didn't before or
Yeah.
didn't think you would until you were exposed to them. And I mean, I think that's my story as well. Maybe the opposite, right? I always wanted to go into science, and here I am now doing marketing, right? And so it's still science, I would say, but maybe a little bit of shift. But
But the science, I think science communication is one of the pending matters that we have. There is a lot of good science communication today, but we still need more. More. We will have to go further and reach more people.
any advice that you would give to, say, anybody that's early in their career or just starting along a research or a lab based pathway?
Well, biology is beautiful. Life is beautiful. Biology is beautiful. Go ahead.
I love it. It's a great reminder, actually.
Mhm.
Another set of questions we ask everybody. What is your proudest moment in your career, and what is your most embarrassing moment in your career?
Well, I don't remember about embarrassing moments. You always break something and you want to hide it, but you have to assume, okay, it was me. I broke it, so I don't remember much. And also, great moments that the eureka things usually don't are not as eureka as many people may think. Sometimes discoveries come slowly, even sometimes when you have something which is okay, I have found this. You don't believe it at the first time. No, it's okay. I have to check. Probably I did something wrong, and I will repeat it. So the real discovery enters slowly. It's not a eureka. I find it.
That's great. Is there anything that runs through your mind, like let's say, I don't know, an experiment or some research isn't going well? Is there anything you think about or any mottos that you have that help you stay grounded?
Well, experiments always seem to go wrong. So you have to uh, check everything and trust and repeat and be patient because the results at the end come. Most of the time, I'm trying to increase the spirit of my students because they come with the results and they say, "Okay, everything is wrong." Everything. Say, "Oh no, don't worry. It's, just keep going, keep going."
That's awesome. I've same with my experience. I have more examples of things that didn't work than
Yeah,
things
yeah,
that did.
sure.
So by far.
Yeah, sometimes you think the perfect experiment, you say, okay, that's great, that's going to work. Never works. Never works at the first time or the second time. This just being patient is important.
Yeah, I would always ask myself too, like, try to ask good questions with some of those failures. Sometimes it's what I like to call user error, where I did something wrong, right? And it was nothing but me that my hands or I miscalculated something, right? But oftentimes, there's something wrong or something that you didn't expect. That it's not the experiment; it's how you approach the question, right?
Even the question, maybe you are not doing the right question. So, and you have to think the way.
That was Dr. Jesús Mingorance, researcher at Hospital Universitario La Paz in Madrid, Spain. This episode of Absolute Genius was produced by Sarah Briganti, Matt Ferris, and Matthew Stock, with more great conversations around the corner in upcoming episodes. Stay curious, and we'll see you next time.